I should point out this, that it is already known how it is that voltage change impacts the transcriptional machinery. People often sort of say that it would be great if we understood how voltage controls gene expression. We basically do. We already know about 6 ways that voltage change transduces down into second messenger pathways and controls gene expression. And this has typical mechanisms like calcium, which neuroscience is very familiar with. Other things like voltage sensitive phosphatases, neurotransmitter transporters. We already know how this works at a single cell level. This transduction machinery has been dissected. In fact, also some of the downstream targets have been identified. So the genes that are controlled by this, all of your favorite BMPs, sonic hedgehogs, FGFs, all of these kinds of things are in fact the redistribution of morphogens in the examples that I showed you. All of this is known both from specific candidates and through unbiased, let's say, RNA-seq and microarray types of things. We know this, but what's interesting is that these answers as to how it works on a single cell level have been fairly unhelpful in understanding the large scale picture that we're really interested in. And it's very much analogous to this multi-scale problem in neuroscience. You can track the pathway, and we do, and in every paper you have to sort of go from the channel to the transduction machinery, which genes are downstream, so you can do all this. But that actually leaves open some much deeper questions about how the collective makes decisions. And so for that reason, we've made the simulator, and this was created by our center member and collaborator, Alexis Pytak. And what it allows us to do is to build models of both molecular networks like GRNs together with the various biophysics steps that happen. And you can simulate every cell having these various circuits. And then you can try to build a full stack model that integrates from the molecular steps to the tissue level physiology, like every cell has various channels. What is the electrical pattern going to be in this tissue? What does that look like on an axial polarity level? You know, where's the head, where's the tail? And then can we extract from this the actual rules of the decision making? Can we turn this into a kind of algorithmic description that looks like this, that makes it much easier for us to control the process? Because now this is almost like a high level programming language where we can see, okay, here are the measurables and here are the decision points that the circuit is using to control large scale patterning modules like make a head, make an eye. And so what we're doing is literally integrating every part of this so that you can build it up and then exert all your manipulations at this level. So you could have your simulator and ask questions like, why does the head-tail gradient rescale when you cut the planarian into 4 pieces? You know, how does that work? And you can't see that by drilling down into single cells. You have to come back out with that information and build these collective kinds of models. And the other nice thing about these models is that they are the perfect fodder for machine learning approaches to do 2 things: to infer better models from data, okay, and we've done some of that, and also then to interrogate those models for interventions. And so here's some software that anyone can play with if you want to download and kind of simulate all this. And we've shown that you can actually use machine learning to find models that are really good at explaining functional data sets. In other words, you know, you did something to the system and then something else happened. What is a model that explains that? And then find these kind of needle in a haystack interventions that are ways in which you could perturb it to get it to do what you want. Okay, so that I think is the kind of the future of using this for regenerative medicine. So let's talk specifically about a couple of applications in the last few minutes. The first thing is again the cancer issue. So I've shown you that you can track the shrinking of these kinds of computational boundaries from that of the organ or whole body scale to single cells, and you can use this as there's an obvious application here as a kind of diagnostic modality, right? It's screening for cancer and so on. But functionally, what's really important is that first of all, you can induce basically a conversion of normal melanocytes. So here they are, these little black dots are pigment cells, they're melanocytes. if you depolarize a very specific cell population in the animal, not the melanocytes themselves, but a different cell population, we call them instructor cells for an obvious reason, those cells tell the melanocytes what to do, in particular to stay nice, cooperative melanocytes under control. If you block their ability to signal, what happens is basically metastatic melanoma. These normal melanocytes convert to these crazy, really kind of long stringy things that start to crawl away. They dig into the brain, they dig into the neural tube, they start to invade. Here they are all through the blood vessels. This is basically exactly like a melanoma type of behavior. And you can see here, these blue cells, these are the ones that we actually depolarized. They are the ones that are now failing to keep the normal melanocytes behaving correctly. And the melanocytes are just, at this point, they're going on their own. So it's a voltage change in the environment. There's nothing genetically wrong with these animals. There are no oncogenes. There were no carcinogens, nothing like that. But this voltage change is a physiological switch away from the cooperation of cells toward embryogenesis and towards single cell behavior. Now, what's cool is that you can override this. So if we inject an oncogene and they make these tumors, of course, they're labeled with the red fluorescent protein. What you can do is co-inject a channel, and you can force these cells to stay electrically coupled to their neighbors, despite what the oncogene is trying to get them to do. And here, while that dominant negative KRAS, P53, whatever, while they're blazingly expressed, there's no tumor. It's the same animal. There's no tumor because these cells are coupled to this electrical network that forces them towards to a proper tissue fate. Okay, so that's where our sort of cancer applications are going, is to, and we've done this with light, with optogenetics and so on, and now we're in human cells to try to use this to target carcinogenic behavior in human cells. The other thing that is important is, again, to think about the level of control that's possible with this. Here is a normal tadpole brain. So you see forebrain, midbrain, and hindbrain. And what you can do is, if you, for example, introduce a mutated Notch protein. So Notch is a very important neurogenesis gene. You can mutate it and introduce a dominant mutation. You can see forebrain is basically gone. Midbrain and hindbrain are just a bubble. These animals have no behavior to speak of. They lay there, they do nothing. You can do the same thing with alcohol, nicotine, various teratogen exposures. What you can do is build a computational model. And again, this is the work of Alexis Pytak, who's a staff scientist in my group and a few collaborators. What you can do is you can build a computational model that asks a simple question: what determines the shape and size of the brain? And there's a particular bioelectrical pattern that's required for the brain. And then on these backgrounds of teratogens or even Notch mutation, you can ask the model a question. You can say, if this pattern is disturbed, such as it is here, what channels would I have to open or close to get back to the correct pattern? And the model told us, and it said there's a particular channel, HCN2, which can help us sharpen these boundaries, even though there are massive defects in the Notch signaling and with these various other pathways. You can still reinforce these boundaries by opening HCN2. And sure enough, if you do it with drugs, or if you just misexpress HCN2, you can get back to a normal brain shape, a normal brain gene expression, and in fact, normal IQ. These animals get their behaviors back and their learning rates back. So here's what this is. This is using a very specific computational model to rationally manipulate the electrical signaling to get back to a very complex organ morphogenesis. And so the whole sort of roadmap looks a little bit like this. We already have expression data, profiling data on which tissues in the body express which channels. So that's already known. It needs a lot more work, which is if you knew what the voltage pattern was supposed to be in various healthy tissues under various conditions. So basically physiomic profiling is needed. Then we have this computational simulation engine, which is able to say, if you want to go from the incorrect pattern to the correct pattern, which of these channels would you have to open and close? Okay, and that tells you immediately what cocktail of ion channel drugs you would need to use. And so we've developed this pipeline. You can play with it here and choose your tissue and so on. And the idea is that something like 20% of all drugs are ion channel drugs. And these things form an incredibly convenient toolkit of what we call them electroceuticals, because if you have the right computational model, you can repurpose these, which, and the human safety data is done on these things. People already take them for all kinds of uses, and they have a massive applicability in tweaking the bioelectrical signals. Now, another nice thing about this kind of approach is that once the bioelectric circuit has made its decision, the downstream steps that are in second messengers, then gene expression, everything else, it can be as long as you want. So for example, in the case of the frog, a 1-day, 24-hour application of a particular cocktail gives you 13 months of leg growth, right? And you don't have to micromanage that whole process. It's just figuring out what electrical state is going to shift the tissue towards a particular goal state that all of the cells are going to work towards. So I'm just going to close here and summarize as follows. There's this really important layer of physiological decision-making that sits between the genotype and the anatomy. And it's becoming a really tractable target for biomedicine and also synthetic bioengineering, because we can now see how the electrical dynamics that are operating in that tissue encode particular anatomical layouts towards which the cells continue to build. And evolution apparently discovered very early on that this electrical signaling is a really convenient medium for computation and global decision-making. It's not an accident that all nervous systems use it, and all of our computer technology uses it. Ion channels, especially the voltage-gated ion channels, are basically voltage-gated current conductances. They're transistors, right? And that's a very powerful architecture that evolution found long before we did for forming feedback loops, memory circuits, and so on. And we think that cracking this bioelectric code can help reveal how cell networks make decisions in large-scale anatomy, not just how individual cells decide what type of cell they're going to be, but actually the real question of growth and form. Where do these complex patterns reside? And we can now rewrite some of these patterns, such as, for example, in the planarian, or in the frog. And new machine learning tools are coming online to help us design strategies for all kinds of applications. Thank you for the opportunity to speak to all of you and to share some work and get your feedback. So if anyone would like more details or to get in touch with me, all the papers, everything else is at these websites here. So I would like to thank the people in my group who did the work. These are all the postdocs and students and others who did the work that I showed you today. Here are all, some of our collaborators who are working with us. Of course, the model systems that we work with, all kinds of animals, and our funders. Very grateful to them. Again, disclosure, Morphoceuticals Inc. is the company for our frog limb regeneration work. And, just in the end, I always like to show a video of these two-headed animals. The first time I reported these data at a meeting, someone stood up and said, "That's impossible. Those animals can't exist." And so now I make sure that I bring a video